Good evening, everyone. Today, I'd like to talk about the history and development of biotechnology, a field that has revolutionized our understanding of life and transformed numerous industries. The origins of biotechnology can be traced back to the early 20th century when scientists discovered how to ferment bacteria to produce chemicals such as acetone and butanol. This discovery led to the development of the first industrial-scale fermentation processes, which were used to produce antibiotics, hormones, and other important chemicals. In the 1950s, the discovery of the structure of DNA by Watson and Crick paved the way for the field of genetic engineering, which involves manipulating the genetic material of living organisms to produce new products or improve existing ones. This breakthrough led to the development of a range of biotechnology products, including genetically engineered crops, recombinant DNA-based drugs, and diagnostic tests for genetic disorders. The 1970s saw the emergence of the biotech industry as we know it today, with the founding of Genentech, the first biotech company, in 1976. Since then, the industry has grown at an unprecedented rate, driven by advances in molecular biology, genomics, and bioinformatics. Today, biotechnology has applications in numerous fields, including agriculture, medicine, energy, and environmental science. In agriculture, biotech crops have been developed to increase yields, reduce the use of pesticides and herbicides, and improve the nutritional content of foods. In medicine, biotech drugs such as insulin and growth hormone have revolutionized the treatment of numerous diseases, while gene therapies hold the promise of curing genetic disorders. In energy, biotech processes are being developed to produce biofuels and other renewable energy sources, while in environmental science, bioremediation technologies are being used to clean up contaminated sites. Despite these successes, biotechnology is not without its controversies. Concerns have been raised about the safety and environmental impact of genetically modified organisms, and the patenting of genetic material has raised ethical questions about the commercialization of life. Thank you for listening to my talk on the history and development of biotechnology. As I mentioned earlier, one of the most exciting and impactful applications of biotechnology is in the field of drug discovery and development. The process of discovering and developing new drugs is a complex and time-consuming one that typically takes more than a decade and costs billions of dollars. It involves a range of scientific disciplines, including biochemistry, pharmacology, and molecular biology, as well as extensive testing and regulatory approval processes. The first stage in the drug discovery process is target identification, where researchers identify a molecular target that is involved in a disease process and that could be modulated by a drug. This can involve a range of approaches, including screening large libraries of compounds, studying the genetics of the disease, and analyzing the structure of the target protein. Once a target has been identified, the next stage is lead generation, where researchers identify compounds that can interact with the target and potentially modulate its activity. This can involve screening large libraries of compounds, as well as using computer simulations and other approaches to design new compounds that are optimized for the target. The next stage is lead optimization, where researchers work to improve the potency, selectivity, and pharmacological properties of the lead compound. This can involve modifying the chemical structure of the compound, testing it in animal models, and optimizing its pharmacokinetic properties. Once a lead compound has been optimized, it moves into preclinical development, where it undergoes extensive testing to assess its safety and efficacy in animal models. This can involve a range of tests, including toxicity testing, pharmacokinetic and pharmacodynamic testing, and efficacy testing in disease models. If the preclinical testing is successful, the lead compound can then move into clinical development, where it undergoes testing in human subjects. This involves a series of clinical trials, typically divided into 3 phases, where the drug is tested for safety, efficacy, and optimal dosing in patients with the target disease. If the clinical trials are successful, the drug can then be submitted for regulatory approval, where it undergoes a rigorous review process by regulatory agencies such as the U.S. Food and Drug Administration, FDA. If the drug is approved, it can then be marketed and sold to patients. Despite the lengthy and costly process of drug discovery and development, the potential benefits of new drugs are immense. They can transform the lives of patients with previously untreatable diseases, save lives, and generate significant economic benefits for the companies that develop them. In recent years, advances in biotechnology have revolutionized the drug discovery process, with new technologies such as CRISPR gene editing, high-throughput screening, and machine learning algorithms enabling faster and more efficient drug discovery. In addition to drug discovery and development, another exciting area of biotechnology is genetic engineering, which involves modifying the DNA of living organisms to achieve specific goals. Genetic engineering has a wide range of applications, from agriculture to medicine to environmental conservation. In agriculture, genetic engineering has been used to produce crops that are more resistant to pests and diseases, as well as crops that have improved nutritional profiles. In medicine, genetic engineering has been used to develop gene therapies for inherited diseases, as well as to produce recombinant proteins such as insulin for the treatment of diabetes. One of the most powerful tools in genetic engineering is the CRISPR-Cas9 system, which allows scientists to make precise edits to the DNA of living organisms. This has revolutionized the field of genetic engineering and has opened up new possibilities for treating genetic diseases and engineering new traits in plants and animals. Another application of genetic engineering is in the production of biologics, which are complex protein molecules that are used to treat a range of diseases, including cancer, autoimmune disorders, and infectious diseases. Biologics are produced using recombinant DNA technology, where the DNA that encodes the protein of interest is inserted into cells, which are then used to produce the protein in large quantities. In addition to genetic engineering, biotechnology also has a range of other applications, including the development of biomaterials, such as synthetic tissues and organs, and the use of nanotechnology to develop new drug delivery systems. One of the most exciting areas of biotechnology is synthetic biology, which involves the design and construction of new biological systems with specific functions. This has the potential to revolutionize a range of industries, from healthcare to energy to materials science, and has already led to the development of new vaccines, biosensors, and biofuels. Despite the immense potential of biotechnology, there are also concerns about the ethical and social implications of its applications. For example, the use of genetic engineering in agriculture raises questions about the safety and environmental impact of genetically modified crops, while the use of gene editing in humans raises questions about the potential for eugenics and genetic discrimination. It is important for scientists and policymakers to carefully consider the potential risks and benefits of biotechnology, and to engage in transparent and responsible decision-making to ensure that these technologies are used for the greater good. Another area of biotechnology that has potential implications for drug discovery and development is cloning and cloning technology. Cloning is the process of creating genetically identical copies of an organism, and it has been used in a variety of applications, from preserving endangered species to producing livestock with desirable traits. One potential application of cloning technology in drug discovery is in the production of monoclonal antibodies. Monoclonal antibodies are highly specific antibodies that are used in a range of therapeutic applications, including cancer treatment and autoimmune disorders. Traditionally, monoclonal antibodies are produced by hybridoma technology, where cells from two different sources are fused together to produce a hybrid cell that can produce the desired antibody. However, this process is time-consuming and expensive. Cloning technology offers a potentially faster and more efficient way of producing monoclonal antibodies. By cloning the cells that produce the desired antibody, researchers can produce large quantities of the antibody in a relatively short period of time. Cloning technology also has the potential to improve drug safety and efficacy by allowing researchers to create animal models that are genetically identical to humans. Animal models are an important tool in drug development, as they allow researchers to test the safety and efficacy of potential drugs before they are tested in humans. However, animal models can be imperfect, as they may not fully mimic the human disease state. By using cloning technology to create genetically identical animal models, researchers can potentially improve the accuracy of preclinical drug testing and reduce the risk of adverse effects in humans. Despite the potential benefits of cloning technology, there are also concerns about the ethical implications of human cloning. The prospect of creating genetically identical humans raises a range of ethical and social questions, from the potential for eugenics to the impact on personal identity and family relationships. It is important for scientists and policymakers to carefully consider the potential risks and benefits of cloning technology, and to engage in transparent and responsible decision-making to ensure that these technologies are used for the greater good. In addition to cloning, another area of biotechnology that has potential implications for drug discovery and development is stem cell research. Stem cells are undifferentiated cells that have the potential to develop into a range of different cell types, and they offer exciting possibilities for regenerative medicine and drug discovery. One potential application of stem cell research in drug discovery is in the development of disease models. By using stem cells to create disease models, researchers can better understand the underlying mechanisms of disease and develop new therapies to target these mechanisms. Stem cells also offer the potential to develop personalized medicine. By using a patient's own stem cells to create disease models, researchers can develop therapies that are tailored to the individual patient's needs and genetic makeup. However, stem cell research also raises ethical and social questions. The use of embryonic stem cells in research is controversial, as it involves the destruction of embryos. There are also concerns about the potential for the commercialization of stem cell therapies and the impact on healthcare equity. As with cloning technology, it is important for scientists and policymakers to carefully consider the potential risks and benefits of stem cell research, and to engage in transparent and responsible decision-making to ensure that these technologies are used for the greater good. Another exciting area of biotechnology is biosynthesis, which involves the production of biochemical products through the use of biological organisms or systems. Biosynthesis has potential applications in a wide range of industries, from healthcare to agriculture to energy production. One example of biosynthesis in drug discovery is the production of natural products. Natural products are chemical compounds that are produced by living organisms, and they have been a rich source of new drugs over the years. Biosynthesis offers a potential way of producing natural products in a more efficient and sustainable way, by harnessing the power of biological systems to produce these compounds. Another example of biosynthesis in drug discovery is the production of recombinant proteins. Recombinant proteins are proteins that are produced by genetically engineered organisms, and they have a range of therapeutic applications, from treating cancer to regulating the immune system. Biosynthesis offers a potential way of producing recombinant proteins in large quantities, which is essential for their use in therapeutic applications. Biosynthesis also has potential applications in agriculture, where it can be used to produce crops with desirable traits, such as increased yield or resistance to pests and diseases. By using genetic engineering and other biotechnological tools, researchers can create crops that are better suited to the needs of farmers and consumers alike. In addition to agriculture and healthcare, biosynthesis also has potential applications in energy production. For example, researchers are exploring the use of photosynthetic organisms to produce biofuels, which could offer a more sustainable alternative to traditional fossil fuels. While biosynthesis offers exciting possibilities for innovation and progress, it is important to carefully consider the potential environmental and social implications of these technologies. For example, the use of genetically modified organisms in agriculture raises questions about the impact on biodiversity and food safety. It is important for scientists and policymakers to engage in transparent and responsible decision-making to ensure that biosynthesis technologies are used for the greater good. In conclusion, biotechnology has come a long way since its inception and has revolutionized many aspects of our lives, including medicine, agriculture, and environmental sustainability. From the discovery of DNA to the development of CRISPR technology, biotechnology has opened up new frontiers in scientific research and innovation. Through genetic engineering, biopharmaceuticals, biosynthesis, and biostatistics, biotechnology has given us the tools to develop new drugs, improve crop yields, and produce sustainable materials and fuels. As the field of biotechnology continues to advance, we can expect to see even more groundbreaking discoveries and applications in the years to come. Finally, I would like to thank you for taking the time to watch this video. If you found it informative and engaging, please consider subscribing to our channel, liking this video, and leaving a comment below. Your support helps us to continue creating quality content and sharing